It is my great pleasure to introduce you Eric Brown, who comes from the Department of Biochemistry and Biomedical Excisions at McMaster University, who is going to talk to us about bacterial chemical genomics, a path of thesis resistance. Thank you very much indeed, Eric, for accepting to be here today.
Thank you, Capello. I Googled KPRK when you invited me to give this talk, and it gave me a Montana radio station. And I thought, well, that's pretty interesting. But it's really a pleasure to be here and tell you about work that we've been doing in the area of the gram-negative envelope. My apologies, there's another title in the proceedings document which alludes to a talk on systems biology, and I'll try to kind of combine the two today if I can. I begin with a slide that really speaks to some of the discussion that Karen Bush just began in the question period around the discovery of antibiotics, which are new chemical classes. So this is work out of the Pew Charitable Trusts, which is after some analysis originally by Lynn Silver, which shows the discovery of new chemical classes and the decade in which they were discovered. It highlights, of course, this golden age of antibiotic drug discovery in the 40s, 50s, and 60s, and perhaps more importantly, it highlights the lack of innovation over the last 3 decades. This is pretty depressing to me as a biochemist who's kind of grown up scientifically during this age and really admire the advances in modern drug discovery. This era of modern drug discovery is underpinned by advances in things like recombinant DNA technology to produce targets at very high purity and amounts to facilitate things like high-throughput screening, genomic studies, high-throughput synthesis, computational advances, structural biology, all the stuff that has been, quite frankly, very successful in other therapeutic areas to discover new drugs, but no new drugs for antibiotics. The problem's complex, and the reasons for failure are manifold, but I think it's illustrative to give it some thought, at least. The golden age of drug discovery, of course, is a very different approach. It's often from a natural product or a small synthetic library, isolated chemical compound that leads to growth inhibition on a dish. The marvelous thing about antibiotic drug discovery over the ages is that if you can get that thing to work well in a mouse, then it often will work well in the human. And so there's a sense, as Karen suggested, that the low-hanging fruit are gone, that we've kind of done all we can do with this sort of paradigm. And so we're very interested in my lab in new approaches to this problem. Among other things, we've been asking ourselves lately, have these reductionist approaches that are modern failed to use new drugs because of the nature of reductionism? And so we've been thinking increasingly about systems biology approaches to try to break that logjam for the discovery of new antibiotics. So we do a variety of things in my group, and I have time to tell you about some systems approaches associated with one of the collections that we often use. And this is the KO collection made by Hirotada Mori in collaboration with Barry Warner to create a systematic collection with a deletion in every single dispensable gene in E. coli when growing on rich microbiological media, a collection of some 4,000. And so what we often do is this kind of thing where we're asking for the interaction systematically of each gene with a chemical probe. We've done this for known antibiotics. We're doing it lots for unknown compounds. But what this looks like is the following, and it's after genetic formalisms first made in yeast, the so-called multiplicative rule, where wild type would grow with an index of 1. If we make a deletion in gene X, it's growing at 70% that. Add a chemical number 1 at some concentration, 80%, and we expect then that a combination of a deletion in gene X and the addition of that chemical will grow with the multiplier, or 0.56. If it grows worse than that, we call that enhancement of the growth inhibitory phenotype that are due to that deletion or chemical. Some call it synthetic sick and lethal. And if it grows better, we call that suppression of the growth inhibitory phenotype. So with those formalisms, then, we do this, of course, in high throughput, and it's really a magical time in terms of systems biology to be able to pin and create colony maps at very high density. And so here's an example of a 6,144 density plate in which the KO collection has been pinned onto a uniwell microwell plate at very, very high density. And now we can put any compound that we choose into this place to ask kind of one gene at a time what the interactions of that chemical with the genes are. Sean French has really been a pioneer in this stuff in my lab. He was helped initially by Hirotada Mori, who first set up a system like this in his lab in Japan. But basically, it consists of transmissive scanners that are in a 37-degree incubator. They scan through the plate and allow one to get a kind of absorbance density for each colony growing. And so we monitor these over time. And as you can see in the animation here, we get through integrating the density associated with that transmission, we can compute a volume for the growing colony over time. I confess we're a little bit compulsive about this in my group, and so at some point, a fellow called Chan Mangat cut out a number of these colonies over time and determined that the colony-forming units, as a function of that integrated density, was actually a straight line. And so that meant that we could actually compute a growth curve for each of these strains over time, growing at very, very high density. And shown here is a collection of 1,536 growth curves. Typically in these kinds of experiments, folks have looked out here at a very late time point to try to get a sense of the fitness of that colony. But we're very interested in this portion of the growth curve, of course. That is the doubling time of any strain. It's a fundamental constant of a particular strain of bacterium, and it's much, much less prone to error as a function of, for example, inoculum effects or edge effects that are common in these high-density experiments. So here's a typical systems biology hairball, of course, where we have the interactions between genes around a collection of antibiotics that fall into various chemical classes: folate, cell wall, DNA replication, protein translation, RNA synthesis. There are both colony size interactions here, that's the endpoint read that I alluded to earlier, as well as the growth rate interaction. In total, there are some 1,500 interactions here for 15 different antibiotics of various chemical classes and mechanisms. So that's about 100 interactions per antibiotic. You get a sense of a very highly connected and redundant system that is trying to resist perturbation, right, by an antibiotic. Okay, so what do we do with this kind of information? We're very interested in this problem, the so-called gram-negative challenge. Another image, actually, from the Pew Charitable Trusts highlights very, very nicely, I think, that of all the antibiotics that we know about, the vast majority of these work well on gram-positive bacteria. Right? And that's, of course, because they're not so good at penetrating the envelope of gram-negatives and resisting the efflux pump systems that are common in gram-negative bacteria. And we've wondered in my lab whether or not there would be value in trying to think about ways to make gram-positive only antibiotics more effective against gram-negative bacteria. And so here's a collection of gram-positive only drugs, of everything from oxazolidinones to macrolides, glycopeptides, rifamycins. And if we could manage to break this barrier in gram-negative bacteria, that we would have now at our disposal perhaps a large number of potential drugs for treatment. So here's a chemical genomic experiment for both rifampicin and erythromycin. So some 140 or so genes that sensitize E. coli to rifamycin when deleted. And so if we look at those that are unique to RIF, and we look at the Go terms that are enriched in terms of the processes that are going on that lead to this enrichment, we're looking largely at lipopolysaccharide synthesis as a dominant method for sensitizing bacteria to RIF. And similar processes for erythromycin. If we look at the intersection of these two, things like protein insertion into the membrane, outer membrane assembly. And this gives us thought, of course, to these sorts of processes. This is how you build an outer membrane in a gram-negative organism. It involves the synthesis of lipopolysaccharide and its trafficking to the outer membrane, the synthesis of outer membrane proteins and their movement across this periplasmic space to the outer membrane. Also lipoproteins, phospholipids. And so this is the process, of course, that we need to think about if we want to try to sensitize a gram-negative bug to gram-positive antibiotics. Okay, so can we screen then for chemical compounds that would target outer membrane biogenesis? This is a bit of serendipity. This fellow, John Stokes, was a PhD in my group, now a postdoc in Jim Collins' lab at the Broad in Boston. But he made this observation. It was a control experiment. He was screening for inhibitors of ribosome biogenesis, which is a cold-sensitive process. And I asked John to use a cell wall antibiotic, and sure enough, he saw cold sensitivity for vancomycin function. So here's growth inhibition by vancomycin with a single-digit MIC at 15 degrees Celsius. 20 degrees, 25, 30, 37, 42. So clearly this is a temperature-dependent inhibition of a gram-negative by a gram-positive only antibiotic. John was careful to put the Van gene cluster, the resistance cluster, into E. coli to show that nothing weird's going on here. This is just about allowing vanco to get to target. He did another clever thing. He looked for suppressors of this process, the suppressors of the cold sensitivity of vancomycin. And he pulled up a bunch of genes from the KO collection that were involved in those same processes that I just highlighted. So paradoxically, suppression of vancomycin function in the cold is in, for example, predominantly LPS biosynthesis. So we knew immediately that that was going to present us with an interesting screen. We're very interested in idiosyncratic interactions like this. We like antagonism for chemical screens. And so the following screen came out of this. It's a screen for life, so we've got E. coli at 15 degrees and an inhibitory concentration of vancomycin, incubate with a chemical library, and if we have an active compound, we'll have a well where we see growth. We like this screen a lot. It's got the outer membrane target list that I said we were interested in. It's based on idiosyncratic genetics. It gives rise to very low frequencies of actives, little or no nuisance compounds, because it's a growth for life. You can imagine it'd be great for natural product libraries, et cetera, and increased sensitivity. I could get into this later if folks are interested. We screened at first in a pilot screen, as we often do, a collection of 1,500 known drugs. And to our surprise, we brought up this particular compound, pentamidine. It's this aromatic diamidine separated by 5 methylene groups. It's an antifungal drug that's used often prophylactically in cancer treatments to prevent fungal infections of the lung. It's also antiprotozoal. It had never before been linked with any sort of antibacterial therapies. And so we were interested in having a close look at this compound. We did a number of tests for its action. We determined that it does not inhibit an enzyme that leads to outer membrane synthesis, but actually interacts physically with the outer membrane and disturbs it. So shown here is an AFM experiment where we're dragging a probe across the surface of E. coli, and we see undulations that are on a nanometer scale, typically for a wild-type bacterium. Incubation with pentamidine, though, leads to deep furrows in that surface and perturbation in that surface that is somewhat reminiscent of polymyxin, of course, which is a known antibiotic that disturbs the structure of the outer membrane through physical interactions. So enter Craig McNair, a PhD student in my group that's been very successful in developing various animal models. Craig developed an Eibomanii model of systemic infection, and he was very interested at the very synergistic interaction between novabiocin and pentamidine. So these are checkerboards that we like to do in my group, where dark is growth and white is no growth, and it shows a synergistic interaction where we have a dose-sparing effect on the amount of novobiocin which is required to inhibit the growth at additional concentrations of pentamidine, suggesting synergy between these two compounds. Pentamidine's taking out the outer membrane. Novobiocin, a gram-positive drug, is getting to target. Craig did quite a bit of work here. This is a survival curve for 10 animals. We dose, and then 2 hours later, treat with novobiocin. The combination of novobiocin and pentamidine led to complete survival of 10 individuals in this experiment. And here's a look at some of the colony counts. This is for the spleen, but it didn't matter the way he looked in blood or lung or liver or kidney. They all look the same as this. Very, very high counts in the control, very little impact with either pentamidine alone or novobiocin alone, and sterilization of this organ in the presence of both of these compounds. So we're pretty excited about this experiment. I mean, it's a success for us, of course, and we've been working to try to make pentamidine even better. It has some well-known tox issues, and we've been working with Deb Hung's group at the Broad through an academic kind of baby CARB-X effort to try to optimize this compound, and stay tuned for the results of those efforts. Meanwhile, we've expanded our screening effort. Right? There was just a small pilot screen of known drugs to generate that active compound, and J.P. Cote, who's a postdoc in my lab, just took possession of his own laboratory recently at Sherbrooke University. He and Maya Farah, a research associate in my group, have been working on this effort. We have a collection of compounds of about 500,000 diverse synthetic compounds at McMaster, as well as a collection of natural products from Jerry Wright's lab. And these two have been going through these collections using our vancomycin suppression assay to look for new chemicals. They've identified some very interesting classes of actives here. So some of these compounds are just generally membrane active. We heard from Bob Hodges earlier in the week that can work, particularly on the inner membrane, but we're trying to stay away from the inner membrane, really focusing on activity on the outer membrane, which we think is a non-canonical membrane that should be possible to achieve selectivity. So we have also some outer membrane selective compounds that we're pretty excited about, and compounds that we think are inhibitors of outer membrane biogenesis. So we're working through these now. I work with a fantastic group of young people at McMaster, and I think I've listed here just about everyone who's ever funded me or whom I have consulted for. I've highlighted most of these folks from my group as I've gone through the talk, but also to highlight the collaborators here, Deb Hung at the Broad Institute around analoging of pentamidine, Maury at the NAR Institute, Chris Whitfield at the University of Guelph, who's an expert, of course, in LPS synthesis and things out of membrane, and Jerry Wright, my longtime collaborator and friend at McMaster around natural products. And thank you very much for listening.
Thank you. Thank you very much, Eric. Very informative. So we have time for questions. I have one, by the way. In the case of the Baumani infection, did you make any studies about the mechanisms of action? Why the combination works well, while individually they do not?
Yeah, so we, you mean in terms of that infection model? Sorry, say that again. I missed the very last bit.
Yes, no, when you talk about the Baumani infection,
yeah,
you have developed studies about the mechanisms of interaction, so how that works.
Yeah, so in that case, quite a bit of in vitro data that, of course, I didn't have time to tell you today, but we're very confident that pentamidine interacts with the outer membrane of Baumannii, that it's now facilitating the entry of novobiocin in a way that novobiocin is incapable of as a large scaffold hydrophobic antibiotic, and I mean, we really see a dose-sparing effect, both in vitro in terms of kind of a checkerboard analysis, but also in vivo. the amounts of each of those compounds was really dose-sparing compared to what is commonly used in terms of the human doses, is how they would relate to the mouse.
More questions? Yep.
Really nice presentation. If you repurpose an old antibiotic or a compound that's used for something else, what's the road to ultimately getting that approved and used in humans? Is that a handoff to somebody else, or do you have the capabilities of doing that in-house?
It's really tricky. In this case, so I think occasionally, a discovery lab can find some methods, intellectual property around the combination, say, of drugs, but that's also not super strong IP in this space. And of course, what everybody wants for commercialization is market exclusivity. In this case, there's just too much prior art around pentamidine for that kind of IP. And so this is clearly an area, I think, for public health to come in. And I think, what it would take, for example, I think for a clinician to use pentamidine and a gram-positive drug in the clinic is surely a trial to demonstrate that it would work. So I think there's a real role for public health. And having heard from Karen about the exits of many in the private sector in this space, I mean, maybe that's a real option around repurposing. And I think we're hearing some of those ideas, particularly out of Europe and GARD-P, et cetera. But I think there are also probably opportunities in this case. Pentamidine's probably not the best compound, right, that we can think of to do that job. There are kind of some known toxicity issues, and we've been thinking about perhaps can we make that compound better, where it's more potent, less toxic, and it kind of gets the job done.
Last question. Yes, over there. Yep.
Have you checked the level of synergy between polymyxin and vancomycin?
Polymyxin and vancomycin. I'm sure we've done that experiment, and I'm sure that it does work. So polymyxin is also very good at disturbing the outer membrane, right, and allowing gram-positive drugs in. In fact, we published a paper about a year ago where we showed that even works in an MCR background. So in other words, modifications that would happen to the outer membrane paradoxically do not impact very strongly the ability of polymyxin to disturb the outer membrane so that gram-positive drugs can get in. I can't cite perfectly right in this instance, how strong that interaction is between polymyxin and vancomycin, but it works for many gram-positive drugs.
Well, I am going to allow 2 questions more. One from this nice lady.
Thank you, Jose. Yeah, thank you, Eric, for your talk and for the students. I've been in Eric's lab. It's just such a great lab. I went as part of my sabbatical, and I wanted to leave my position and stay there. So it's amazing. It has all the cool tools. So my question is regarding your target, the outer membrane, right? And I can see, well, it's the most external part of the gram-negative. You don't need to worry about permeability a lot. But it's also highly variable. So are you looking for targeted molecules that target specific pathogens? How do you see that in the future?
Yeah, I think you're quite right. I think it's very likely that chemicals that do this will have relatively narrow specificity. Honestly, we're pretty surprised with how many gram-negatives that pentamidine worked on. I didn't have time to show you the data, but it seems to work on a wide variety. And that's probably because it's interacting, with, say, negative charges that are dominant on most outer membranes and many gram-negatives. But I think you're quite right that as we flesh this out some more, I'm guessing that we're going to see some real species selectivity.
Logic says that the pentamidine is positively charged, as you've just said, interacting with negative charges on the lipopolysaccharide in the sort of way that polymyxin does. And you say, and logic again supports this, that polymyxin should permeabilize the outer membrane of gram-negative bacteria. And yet, the data now coming out is that polymyxin combinations, even with anti-gram-negative agents, which should help get in, are significantly inferior to newer beta-lactamase inhibitor combinations. How confident are you that this permeabilization model is going to work and really get things in usefully in the clinic when we're seeing that polymyxin combo just don't work as well as one think that they should?
Yeah, I mean, so polymyxin, of course, is a really marvelous and maybe still enigmatic mechanism of action. But what it does is it facilitates its own entry, and its real antibiotic effect comes from its action actually on the inner membrane, not on the outer membrane. So we might posit that polymyxin also is not the perfect outer membrane permeabilizer. It suffers, of course, from all kinds of toxicities that surely relate to its action on a canonical phospholipid membrane, right, the inner membrane, that you would argue that it might share to an extent with the host. So I think the jury's out. I'm trying not to make any predictions either, David, but I think it's truly worth some effort to explore.
It's a fascinating area. How do the doses of pentamidine that you're having to use for this putative permeabilizer effect compare with those that would ordinarily be used with pentamidine for its present clinical uses? Are you at the same sort of level, significantly higher, significantly lower? I appreciate so far you're in rodents, you're not in man.
Yeah, no, it's great, yeah. So yeah, we can think about sort of the mouse dose relative to the human dose. We're thinking scale of surface area of the organism. The doses that we gave of pentamidine are about 20% for what you would give typically for a systemic dose of pentamidine in a human. So that is kind of promising, right?
So
that we can spare
to
extent the dose and maybe the tox.
Well, we thank the speaker again. Thank you very much indeed for this nice talk, Eric.